Hey friends, in this video we are going to study about elongation stage of DNA replication, and in the previous video we have already covered the initiation stage of replication. In the elongation stage we mainly study that how the elongation or the formation of new strands of DNA take place. So here we have this diagram and it shows the two antiparallel parent strands of the DNA. We have MCM complex that helps to break the hydrogen bond and act as helicase. Here we have topoisomerase 2 that helps to release the tension. These are the RPAs, which is replication protein A, that act as single-stranded binding protein which were present in prokaryotes. They helps to prevent the re-annealing process. Now we see that we have two different polarities of the parent strand. One is the 5' end, other is the 3' end in the replication fork. Now because of this different polarities, RNA primer is recruited in the different positions. We know RNA primer can be recruited from 5' to 3' direction only. Why from 5' to 3' direction only? The reason is that 3' end is a free end. It is having hydroxyl group, and this hydroxyl group actually attacks the deoxy adenosine triphosphates, and it can be adenosine, it can be thymine, it can be cytosine, it can be guanine, anything, any of the nitrogen base which is present in the form of triphosphate. The 3' end having hydroxyl group attacks on that, nucleophilic attack take place, and then those triphosphate breaks into monophosphate. So here we see these triphosphate actually breaks into monophosphate, and this further PPI undergoes hydrolysis in the presence of enzyme pyrophosphatase and releases energy. This energy helps to establish the new hydrogen bonds between the new strands of the DNA. So here we see this is the reason why the RNA primer is recruited in 5' to 3' direction only. So because of this kind of recruitment and because of the different polarities of the parent strand, RNA primer is recruited, one in the free end, another is in the fork end. And over here we can see that here we have the 5' polarity. So because of the 5' polarity of the parent strand, RNA primer cannot be recruited at the free end, and because of this reason it has to be recruited multiple times in this direction. So because of that, multiple RNA primer is present in this strand. So this strand is known as the lagging strand, and this lagging strand forms the fragments of DNA which is not attached to each other and are not continuous, so they are called as the lagging strand, and these fragments are called as the Okazaki fragments. Over here we can see this is the leading strand, because we have recruited the RNA primer from 5' to 3' direction only, and this helps in the formation of new complementary strand very easily. So we can see that in the elongation stage we get leading strand and the lagging strands of the new DNA. Over here now we have enzymes that plays major role in the formation of these strands. In this leading strand we see that DNA polymerase eta takes place, that helps in the polymerization, while in the lagging strand we see DNA polymerase delta. This DNA polymerase delta and eta, they are having the similar kind of structures. They are actually having a clamp which is called as the pCNA ring. This pCNA illustrates for proliferating cell nuclear antigen. This ring is actually a clamp that holds the DNA, that holds this parent DNA and helps to recruit new nucleotides by base pairing. Now this pCNA ring is actually loaded by a loader which is the clamp loader called as the RFC. RFC is replication factor C. Replication factor C, it's a clamp loader that brings the pCNA ring to the parent template. So this parent DNA or the template. So this is present in both DNA polymerase eta and DNA polymerase delta. So this ring helps in the polymerization step. And we know that in the previous video we studied that RNA primer is itself is brought by DNA polymerase alpha, because DNA polymerase alpha over here act as primase, as well as it also helps in the recruitment of some of the nucleotide after it. So after this, because of the low processivity of DNA polymerase alpha, the new polymerization enzymes needs to be recruited. So we can see that in the leading strand we have DNA polymerase eta, and in the lagging strand we have DNA polymerase delta. Now this is done. Other thing that we can notice in this is that we have over here leading strand and we have over here lagging strand. The leading strand is continuous and lagging strand is discontinuous, hence this is a kind of semi discontinuous process. Other thing that is we can observe is that one is the leading going in this direction, other one is going in that direction. So this is actually a bidirectional process. Other thing that we can notice is that when the new DNA strands are formed, the one of the DNA remains the parent DNA while other strand is the new strand. So this is a kind of semi-conservative also. So here we can conclude that the replication process is bidirectional, semi-discontinuous and semi-conservative. This thing we can observe in the elongation stage very easily. Now since we have seen that both these strands have been formed, now the major role is that we need to remove this RNA primer, because it can't be there for the long time. So RNA primer needs to be removed by a enzyme which is called as RNase H in the leading strand. So in the leading strand what we see is that RNA primer is removed by RNase H, but in the lagging strand these primers are actually removed by FEN1, which is called flap endonuclease 1. So flap endonuclease 1 removes the RNA primer in case of lagging strands. And when why flap endonuclease 1 is used in lagging strand, the reason is that when this new polymerization or the new chain is synthesized, or this new strand is synthesized, it pushes the RNA primer to some extent, and because of this push it becomes a hanging part, and that hanging part needs to be removed. This is very clear through this diagram, that we see that when this new strand is forming, it actually pushes this one 5' end, so it pushes this end, and this leads to the hanging of this RNA primer. So this hanged RNA primer needs to be removed. This hanged portion is called as flap, which is removed by FEN1, flap endonuclease 1. And when this is removed, these fragments they get continuous, and since there is gap left between them, this gap is actually filled by DNA ligase, and then it forms a full complete new strand of DNA. So this becomes a whole. This is the whole process how it actually occurs. So this is how the polymerization step takes place and an elongation occurs. Now what we see that after the formation of both the strands, which is leading and lagging strand, they both are formed and RNA primer is removed. In this case, one thing that we need to remember is that in leading strand it is removed by RNase H, while in the lagging strand the primer is removed by FEN1, which is flap endonuclease 1. So after they get removed and the gap is filled and they are sealed, so now they needs to be packed. So they packed with the help of histone proteins. So there are certain factors that is required for the packaging of this newly formed DNA with the histone proteins, and these factors are actually NAP1 and CAF1. NAP1 is the nucleoid associated protein which brings the histones inside the nucleus, and CAF1 is the chromatin assembly factor 1. It allows the DNA to wrap around the histone. So these two proteins or these two factors they play important role in compaction of the newly formed DNA. So this is the elongation part of DNA. Next, what we will study is the termination of DNA, which is a little complicated process as compared to prokaryotes. Because of the presence of excess primer, because of these multiple removal of this primer is not as easy, and this leads to the problems in the telomeric region, because the termination part actually takes place in the telomeric region of the chromosome. So this is a kind of different process. So I hope that you understood this process of elongation of the DNA in eukaryotes. I hope you liked this video, and if you did, then please don't forget to like, subscribe, share, comment. Thank you.